In 2000, scientists from the Human Genome Project announced that they had successfully sequenced the human genome. However, the human genome is somewhat of a misnomer. Our genome is comprised of about 3 billion base pairs, and although we share most of our nucleotides, small variations exist in the form of single nucleotide polymorphisms, copy number variations, and others. Furthermore, humans have another genome, the mitochondrial genome. Our mitochondria, organelles in our cells that provide energy for us, also have their own genome. A single nucleotide polymorphism, or SNP, is a base pair in the genome where, across the population, multiple nucleotides, A and G in the example on the slide, are present. Importantly, SNPs are inherited. Therefore, individuals who are more related to one another will have more similar SNP profiles. Historically, molecular anthropologists have studied SNPs in the mitochondrial DNA, which is passed from a mother to her offspring, and on the Y chromosome, which is passed from a father to his sons. As shown in the diagram, children only receive mitochondrial DNA from their mothers. This is because only eggs carry mitochondria. Studying mitochondrial and Y chromosome DNA can yield information about ancestry and help anthropologists determine haplogroups. Today, direct-to-consumer genetic testing is extremely popular, and many people are learning about their backgrounds and haplogroups from companies like 23andMe. For this project, DNA was obtained from 23 students over the course of several years. This was done using a buccal or cheek swab. The mitochondrial DNA was sequenced, and the results are presented as lists of mutations relative to what is called the Cambridge reference sequence. Note that the mitochondrial DNA genome is very small, about 16,000 base pairs, compared to the total size of the whole genome. The mitochondrial DNA genome has two main regions, the coding region, where genes produce functional proteins, and the non-coding region, which has no genes and serves to regulate mitochondrial DNA transcription. This project focuses just on mutations in hypervariable regions 1 and 2, which are located in the non-coding region. These regions mutate more quickly, accumulating more SNPs, and are therefore more useful in looking at small genetic differences between individuals and populations. Next, the data is processed to produce a network. The network arranges the individuals, or unique sets of mutations, in a two-dimensional layout based on similarity. However, the first construction contains multiple reticulations, or geometric forms in which the network shows alternative paths between the mutations. In other words, the data we put in is not sufficient to resolve these alternative branch patterns. To resolve these reticulations, the network can be reconstructed after weights are assigned to various mutations. Mutations are typically downweighted if they are known to be less predictive because they are recurrent, or because of other factors. After a better network is produced, the individuals can be assigned to appropriate maternal haplogroups. A haplogroup is a set of mitochondrial DNA SNPs that occur together and correlate with a geographic origin. Haplogroups are definable because SNPs accumulate in historical sequence due to a lack of recombination, which creates different branches that reflect the temporal and geographic context in which they arose. Therefore, we can trace human movements by following the accumulation of mutations defining haplogroups in different human populations. Haplogroups are assigned by comparing an individual's sequence to the mitochondrial DNA phylogenetic tree and seeing where the individual sits on the tree. This can be done by hand or by using a computer program such as Haplograph. To review, we started with 23 students and, from mitochondrial DNA sequencing, produced 23 unique sets of mutations relative to the Cambridge reference sequence. From these mutation sets, we generated a network to visualize the individuals in two-dimensional space based on similarity. We also assigned haplogroups, or maternal ancestry categories, to each individual. As you can see on the slide, we observed significant diversity in haplogroups among 10 students. I learned a lot from conducting this project. First, I got hands-on experience with an early method that scientists use to study the ancestry of humans. I realized that we can deduce ancestry, or haplogroups, from a relatively small portion of our genome. I was also surprised at the amount of diversity observed in the sample. Although concentrated in Europe, as one might expect at an American university, Penn students produced a broad range of maternal haplogroups from all over the globe. This work was a continuation of lecture discussion and a class project assigned in Anthropology 246. Special thanks to Professor Scher for not only teaching the course but also providing additional help in completing the project and the presentation.